Hey, it's a DNA extraction lab extra. It's me, Moser, and after watching this video, you should be able to describe the steps in extracting DNA, describe what extracted DNA looks like, and explain what information DNA extraction actually gives you. Let's go. So this video is designed to go along with the DNA extraction using fruit lab that we use in my classroom. If you don't have a paper copy of the lab, you can probably get most of the procedural steps and questions from this video. You're going to need bananas, table salt, some dish soap, cold rubbing alcohol or ethyl alcohol, and cold water. You'll also need a plastic bag, a little strainer, some kind of wooden stirrer, and a test tube or small glass. So you should read the pre-lab discussion, if you're doing this for my class, and answer these questions on your own. There are also 2 questions that you'll need to answer after you read the lab procedure. Step 1 of our lab is to separate the cells and dissolve the cell and nuclear membranes. So we've got our banana, we've got salt, dish soap, and cold water. And we're going to place all of these items in a cheap Ziploc bag. Doesn't get a whole lot easier than that. Once we've placed all these items in our Ziploc bag, we'll mush it up with our hands. Ew! You don't want too much soap or too much water, so your product should look about like this. Once you've got it thoroughly mushed, you're going to set a timer for 5 minutes and let it do its thing. The soap is going to break down that cell membrane and nuclear membrane that are made of phospholipids. Remember, cell membranes are made of a fat substance. And the physical mashing is going to pop cells open like little water balloons. Cool. You're now going to strain the cell soup through a funnel into your test tube, or if you're doing this at home, some sort of small glass dish. The detergent is going to isolate the cell membranes and nuclear membranes just like it would the grease in a baking pan and sort of separate them. What will this look like? Well, there's us straining it, and when we get it into the filter, it's going to look like that. It's kind of, gross. Yeah, it is. Now we're going to take that ice cold alcohol, either rubbing alcohol or ethyl alcohol, and it's really important that it be cold. I actually keep my bottle of ethyl alcohol in the freezer in the science supply room to keep it cold enough, and then bring it out only on a bed of ice. Once you've got that cold alcohol, you can tilt your test tube and very slowly add enough to get about a maybe 2 centimeter or 1 inch layer of alcohol sitting on top of the other material. That's going to look something like this. And you can actually see that it's sort of looking fuzzy in there. There are these weird little strands of goopy, snotty stuff rising up into the test tube. I wonder what that could be. It's DNA! And if you use a wooden stir rod, you can actually dip down into that alcohol layer and you'll see all these chunks floating in the alcohol. That's DNA. It looks like snot on a stick. Ew. It does. And unfortunately, it doesn't give us any information. Looking at this gob of snot, if we had a similar gob of snot left at a crime scene, we couldn't tell who it came from. We can't even tell if this is banana, human, dog, maple tree, or earthworm DNA, because DNA all looks the same. We can't tell if this banana was related to another banana on the same tree using this, because again, DNA extracted all looks about the same. If we wanted to get more information about the banana in question, we would have to do a gel electrophoresis to find its nearest relative, or do a full DNA sequence to get information about its variety, any genetic diseases it might have, and other interesting bits. That's it. You should be able to answer all the questions in the lab packet using this video, And as always, if you have questions, just ask.